Hello. Alright. Control room, I am live. Question is my lecture live? Ah, there it is. Okay, welcome to another lecture. This lecture is on Clade deuterostomia. Clade deuterostomia. Whoa, hey, come back here. Now, remember that there is a difference between clade versus classification. All right, classification is kingdom, phylum, class, order, family, genus, species. Clades are groupings of organisms based on relatedness. So, clade deuterostomia, not Linnaean, Phylum Echinoderma, Phylum Chordata. So, we continue. Deuterostomia is characterized by deuterostome embryonic development. So you start with your single-celled zygote, the first diploid cell of the generation, and it undergoes division, multiple even cleavages, until it becomes a hollow ball of cells called a blastula. The blastula being characterized by that hollow area called a blastocele. What makes deuterostomia a deuterostome is gastrulation. So remember, during embryonic development, the first external thing to appear is the blastopore. That's part of gastrulation, the process by which you'll eventually develop a digestive system, right? And in deuterostomes, this first embryonic pore is destined to become the anus. It is the mouth in protostomes. So in deuterostomes, they develop anus first, and then on the other side, you get a little pore, and then eventually you get the connective tube that will represent the complete digestive system, and you have a mouth and an anus. So, deuterostomes all come into the world anus first. So, the first thing that becomes apparent in humans is an anus, and some humans just don't go much further than that when it comes to developing. Okay, we're starting off with phylum Echinodermata, the only deuterostome phylum besides Chordata. Echinodermata, besides rhyming with Hakuna Matata, which means it will now forever be in your head, right? Echinodermata. Anyway, means spiny skin. So, echinos is Greek for spiny derma skin. This includes sea stars, right? So where's my pin there? Okay, so this includes sea stars. This includes urchins. This includes sand dollars and sea cucumbers. You are more closely related to a sea cucumber than a squid. All echinoderms are marine, so they are all saltwater organisms. There's only about 6,000 living species Some echinoderms are in shallow water, like tide pools and the wave zones. Some are extremely deep. You can find echinoderms in the deep benthic parts of the ocean. They're all deuterostomes, of course, or we wouldn't have them in this clade. And they are characterized by an endoskeleton made up of ossicles. So, an endoskeleton made up of ossicles. What does that mean? An ossicle is a calcified plate, and it is subdermal. So underneath the skin of a sea star, right, there's some arms. So if we were to cut this sea star in half, whoa, hey, come back here. Yeah, went away. Give me that screen back. So if you were to just cut this sea star in half, right, underneath the skin would be interlocking calcified plates. And it's the ossicles that are visible in, say, a sand dollar or a dead starfish. When you dry them out, you leave the ossicles behind. So the spines are, like on some starfish and on sea urchins, the spines are extensions of the ossicles. Now, because they are underneath the skin, these are an endoskeleton. The support and protective structures are underneath the skin in the body. So this is an endoskeleton. Now there's some really fascinating things in here unique to echinoderms. And the big one is the water vascular system. Water vascular system is the system by which echinoderms can do things like maintain certain shapes or positions, and also by which they can move and grasp. The water vascular system is hydraulic, right? It sucks water in and moves water through a series of tubes. They do not have a circulatory system, right? They do not have a heart. They don't have blood. They circulate dissolved oxygen in the water directly through their water vascular system. So, respiration, oxygen exchange is done via the water vascular system. Excretion of wastes is done by the water vascular system. How it works, this is a picture of sort of a standard starfish body with 5 arms. We have an intake right here called the madreporite. So water can enter the water vascular system from the madreporite. From there, it circulates around something called the ring canal. The ring canal surrounds the main visceral mass of the echinoderm, where we find the nerves and ganglia, where we find the digestive system. So that's surrounding the circular canal surrounds that viscera, right? So from there, we move from the ring canal, the circular canal, to the radial canals. Radial canals are present, if your echinoderm has them, in each arm. So, radial canals come off of the circular ring. Attached to the radial canals are ampullae. If you have ever looked at a starfish, it has suckers, little sucker feet. And it can extend those little sucker feet, and it can move them around, and it can suck onto things to hold them with its little sucker feet. And basically, the ampullae is a little bulb of water connected to the radial canal and then connected to the tube foot. And if you push water into the ampullae, the water pressure pushes the foot out and it extends the foot. And then to pull the foot in and grasp or suck onto something, you just push water in the opposite direction, send water back into the radial canal. So it's hydraulic pressure in the water vascular system that allows a starfish to move its arms. With this long radial canal, if you flush water into it, you'll generally get that curving arm that curves upwards. So that's kind of cool. So it's all about manipulation of water in the starfish. So water enters the starfish through the madreporite, and then it goes into the ring canal, the circular canal there, and then from there it enters into the arms if present and the ampullae. So pretty neat stuff. After that, we have excretion and respiration. So the coelom of the starfish is actually continuous with the tube feet. And papilla at the end of the tube feet, right? So if we have our tube feet, at the ends of the tube feet, I'm just going to like conceptually put some little extensions here. You'd need a much closer image. Well, actually, I have a good picture of papilla. So, right here, let me clear out the image. Come on. Where'd my little cursor go? PowerPoint conveniently makes the cursor near invisible. So at the tips of the tube feet are these papilla. And these papilla are where gas exchange occurs, where what you think of as the gills are. And so respiration occurs here, and you can get oxygenated water into the tube feet and into the water vascular system. Oxygen just diffuses across the papilla into the water vascular system, and oxygen diffuses from the water vascular system to the tissue. So there's no specific respiratory organ beyond water vascular system. Circulatory, locomotory, feeding, grasping, and respiration. The water vascular system does it all. I love it, man. So here is a standard sea star, right, with a little bit of a cutaway for the anatomy. So we can see there's this big central mass here. This is where the stomach is. Glands that secrete digestive fluid into the stomach are located in the arms. So we produce our digestive fluids in the arms and secrete them into the stomach. So we go from the digestive glands and secrete our digestive fluids into the stomach. Sexual reproduction structures also located in the arms. The digestive tract of a sea star is very short. It is technically a complete digestive tract with a mouth and an anus, with the anus exiting dorsally on top and the mouth being ventral at the bottom. What's neat, so you have your gastrovascular cavity. There we go. So big old stomach. And then off of that, we have our digestive enzymes, right, being secreted by our digestive glands. Right, so we've got our stomach, our mouth, and our anus. Right. Okay. Now, to feed, a starfish mouth is really too small to take in any real nutrients. So, what happens is the mouth opens up a little bit. There's my pin. Come here. I have a 2-monitor setup and I made it onto the other monitor. Oh, that's going to be annoying. Come here, mouse. Gotcha. Haha. Okay, so the starfish mouth opens and the stomach is turned inside out. Right? So it everts its stomach out of its mouth such that the inside of the stomach is now in direct contact with whatever the starfish is eating. So it pushes its stomach out of its mouth so that the inside of the stomach is now going to secrete digestive fluid onto what it's eating. This is useful because starfish are famous for eating bivalves, right? They have to use those strong arms with all those tube feet to pry a bivalve apart. There we go. There's some sweet arms. And then once it's pried apart, they just invert their stomach into there. And then those digestive glands, those big old workhorse digestive glands, secrete digestive fluids into the stomach, out of the stomach into the clam. And it liquefies its meal while it's holding it. It's kind of cool. So, directly absorbs the nutrients from the prey. So a starfish does not swallow anything. It pushes its stomach out, inside out, onto the prey. Like, for instance, if you've got your clam, and it just pushes its stomach into the clam, and then the digestive juices fill the clam up. And then, of course, your clam gets digested and dead. Reproduction. Okay, now, Clade bilateria means animals with bilateral symmetry. Sea stars do not have bilateral symmetry. They have what we call pentaradial symmetry, which means we tend to have 5 or multiples of 5 arms coming off of a central disc radiating outwards. So why would we put these in Clade bilateria if we see radial symmetry? And the answer is their larvae. Their larvae are bilaterally symmetrical. So the larvae are bilateral. Only the adults exhibit radial symmetry. They used to think that the larvae, when they'd catch plankton and do surveys of the organisms in there, they used to think that the larvae were actually different species from sea stars because, quite frankly, looking at the image, they don't look all that much like an adult sea star. All right, sea stars are champions of regeneration. So they are champion regenerators. Wow. That is just a terrible image, or terrible writing. But basically, as long as you have a section of the central disc, right? So if you take a sea star, with its pinoradial symmetry, there's the central disc. If you cut it and there's a section of central disc, you will regenerate a sea star. All they need is a portion of their central disc, and they can regenerate the whole sea star from it. Which means that if you take a sea star and cut it in half at the disc, before long you have 2 sea stars. If you take a sea star and chop it, like a pizza into 5 pieces, you'll have 5 sea stars, as long as each has a sufficient portion of the central disc. There's actually a huge economical problem for oyster farmers. The oyster farmers in the U.S., they catch oysters that primarily live in the kelp forests. The kelp forests have a predator, a top-tier predator who basically maintains the entire kelp forest, and that is the California sea otter. California sea otter eats an awful lot of sea urchins, an echinoderm. Now, California sea otters, unfortunately, also have extremely soft, supple, fluffy fur. And so, right there with the oyster farmers were the fur hunters. And the California sea otter was nearly driven extinct. And what oyster farmers discovered is that when you extirpated a sea otter from its kelp forest, the sea urchins overpopulated and started devouring the entire kelp forest, turning it into a barren wasteland where no oysters will live. Now, instead of trying to figure out how to get more sea otters back, the first thing that they did was massive urchin harvests, in which they took all those sea urchins onto the deck and killed them by chopping them into pieces and then throwing the pieces back into the water, which, caused the urchins to regenerate and way over, and now you had like a doubling or tripling of urchin population. So, as it turns out, the only good way to keep a healthy kelp forest for an oyster farmer or for anyone is to have California sea otters. Okay. One of the artifacts of being a champion regenerator, as long as you have a portion of the central disc, is that you can reproduce asexually. So, a sea star is just the easiest shape to draw here. A sea star can undergo splitting, in which it actually splits itself in half down the middle and then regenerates 2 sea stars out of this. So splitting is an asexual method of reproduction amongst the sea stars. This does not mean that echinoderms, I keep saying sea stars, but this is true of pretty much all echinoderms. Echinoderms can also undergo sexual reproduction, in which they do external fertilization. Good old ocean spray and pray, in which the males release a cloud of gametes and the females release a cloud of gametes into the water around them, and hopefully some sperm meet some eggs. Another class in Phylum Echinodermata is, let me go through the classes in Echinodermata. We have Asteroideae. Asteroidea is honestly what you probably think of. Sea stars. Sea stars and sea daisies. This is a sea daisy. Aw, pretty. In the same class with starfish. We also have Crinoidea. Whoa, hey, come back here. Crinoidea are the brittle stars. Brittle stars tend to have a very delicate appearance. Sea lilies and feather stars. This is the echinoderm that we find as far back in the fossil record as is possible. We find echinoderm crinoid fossils going back 500 million years. That is a terrible, 500 million years. So Crinoidea is the most ancient of the echinoderm classes. Echinoidea. Echinoidea includes sand dollars and sea urchins. If you've ever wondered what a living sand dollar looks like, this is a sea urchin, and a living sand dollar looks an awful lot like a very small sea urchin. So they have colorful skin and then little hairs coming off of them. They're very pretty. So Echinoidea, sand dollars, and sea urchins. Then we have Holothuroidea. Holothuroidea characterized by the sea cucumbers. Ooh, pretty. And then we have Ophiuroidea. I think I made myself a mistake earlier. I meant feather stars when I said Crinoidea. That is a terrible mistake on my part. So we're just gonna write it again, and it has to stand because this is live. Whee! Feather stars. Terrible me. I'm a bad person. Okay. So, last up, Ophiuroidea. Really easy to pronounce and easier to spell. This is the dang brittle stars. I hate myself right now. Let's get past this slide. All right, so here's a nice little summary figure of all of the protostomes that we went through, right? Including our platyhelminths, rotifers, the brachiopods, cycliophorans, molluscans, annelids, the nematodes, and the arthropods, right? Then we also did the cnidarians and the poriferans, and then we have good old deuterostomia, and we have some nice shared derived characters to put in here for chordata. Chordata is really fascinating. Chordata is organisms with 4 distinct characteristics. We'll come back to this slide with all this beautiful stuff on it shortly. All right. Chordata. 4 distinct characteristics in chordata. One is, oh, hey, this is just my roadmap. Good, I can get through this. So we will be talking about the 4 characteristics Then subphyla Cephalochordata, the lancelets that have a beautiful image of all four characteristics. Urochordata, the tunicates, which only show all four characteristics during their larval stage. Then Craniata, the hagfish, where we first see a skull. Then lampreys, agnathans, jawless fish, right? And then we have the jaw. Everything that has a jaw is a gnathostome. And we'll talk about chondrichthyes and more. So first, the four major characteristics. One is a flexible cartilaginous notochord. Come back here. Okay. So, a flexible rod called the notochord. It's long and it runs along the dorsal surface. So, runs along the dorsal body of the organism. Right? And with the notochord running along with it, we see a dorsal hollow nerve cord. The dorsal hollow nerve cord is a major feature of the chordates. Up until now, everything had its ganglia, right? If it had its ganglia, it had its little ganglia, maybe anterior, why not? Here's a little crustacean or something with a pair of anterior ganglia, and then you have them come off and then run ventrally along the body. Ventral nerve cords are how protostomes do. Whereas chordates have whatever nervous system they're going to have, and the nerve cord runs dorsal. Right? So a dorsal hollow nerve cord. The reason it's hollow is because during embryonic development, it develops as a little crest. They call it the neural crest. So at the surface, you'll see a little crest develop, like that. Then as embryonic development continues, the crest gets larger, and the two sides tend to get closer together like that. And then the crest submerges until you have a dorsal hollow nerve cord. So that's the dorsal hollow nerve cord. The notochord is only present in visibly in a few of the clades of chordates. A long flexible rod. In most chordates, it has become a part of the skeletal system. In humans and anything else with a spine, the notochord is the intervertebral discs. So you have your vertebra, and between the vertebra are these large hunks of fibrocartilage that pad them so that they don't smash each other together and grind the bone to dust. And those intervertebral discs are remnants of the notochord. So if the animal has intervertebral discs, those are remnants of a notochord, which is pretty cool. Um, in most invertebrates, you find a rod, but it's solid ventral, and it is not at all a nice character. What am I trying to say? Just ignore me. Getting off track. I need some coffee. That's what I'm going to do. Okay. Notochord. Long, flexible rod. Where'd that go? Come back here, slides. Long, flexible rod. Dorsally located. Dorsal hollow nerve cord. Right? Hollow being a new feature. Dorsal being a new feature. Pharyngeal slits. Pharyngeal slits. Also sometimes called pharyngeal gill slits. Not always used for gills for respiration, but these are pharyngeal slits. All right, the pharyngeal slits allow water to enter the mouth and not pass into the digestive tract. So if you're a little aquatic organism and you're pulling water into your mouth, H2O in, you don't want to swallow all of that water. So chordates have this nifty pharyngeal gill slits where H2O comes in and then can exit before entering into the digestive system. So the gill slits, pharyngeal slits, allow water to enter through the mouth and pass out of the body so that it does not enter the digestive system. This is where we find gills in chordates. Some chordates use these for filter feeding as well. And in chordates that don't actually have slits, like us, right? You see the gill slits appear, the pharyngeal slits appear during embryonic development, but then they fuse into other structures, like the ear. So, finally, we have a muscular post-anal tail. So, if you think about a crustacean, like a lobster, here's some fins at the tail, right? The anus goes all the way to the tip of the tail. Right? If you have an invertebrate and they have a tail, the anus ends at the tip of the tail. In chordates, the tail extends past the anus. So it is a muscular tail, skeletal elements for movement. So pretty cool. Do I have my gecko still? Oh, I forgot about him. Sorry, buddy. Here. Okay, these are the 4 defining features that tell you have a chordate. Chordate. Oh, notochord, dorsal hollow nerve cord, pharyngeal gill slits, and muscular post-anal tail. All chordates show all 4 characteristics at least at some point during development. Let's get into the chordates. We are in phylum Chordata. Now we have subphylum Cephalochordata. All cephalochordates show all 4 features as adults. So, very easy. They are characterized by the lancelet being the big defining species here. Lancelet is a very small organism, very short, lives and burrows into the dirt, and then filter feeds with these little cirri, these tentacles. Not that you need to know that, but if you look at a lancelet, you have your dorsal hollow nerve cord, you have your notochord, you have your pharyngeal gill slits, and you have your post-anal tail. All 4 features right there. Nice. So these are found in the oceans, the southern oceans of North America. There's about 32 species of cephalochordates. It's pretty nice that they retain all 4 characteristics because it provides us a nice generalized diagram of a chordate. So, like I said, they sit burrowed into the sea floor with those cirri for feeding, and they filter feed. They flush water in through the mouth and out across the pharyngeal gill slits in order to get their foods. Continuing forward, we have phylum Chordata, subphylum Urochordata, the tunicates. Tunicates are so named for their adult form, which is very simple, right? Of the 4 characteristics of the chordates, the adult form has pharyngeal slits. And that's pretty much it. They're hollow, and they have an in-current siphon that pulls water through their mouth. Then they can flush water out through the slits and filter food particles so that they can then enter the digestive system and then out through the anus and the excurrent siphon. The adult is just sort of like a hollow hunk of pharyngeal slits. Very simple. So adults are simple. They're sessile. They are sometimes called sea squirts. If you've ever been to the coast and walked around at low tide, those areas of kelp, sometimes you can sort of stomp the ground near a sea squirt, and it will shoot out the water that it has collected. Dang, the microphone is not in good condition. Just have to lay it there. So, adults show very few, if any, of the characteristics. Then we have our larvae. The larvae still retain all 4 characteristics of chordates. So a larval tunicate, so after external fertilization, shooting sperm and eggs out into the water, they get fertilized and the larva hatches from the eggs, and they're a very simple organism, but they have all 4 characteristics. Looks similar to a lancelet, except the mouth is still a siphon. Here we see the tunic, by the way, of a tunicate. That big hollow cavity where the pharyngeal slits are is made of cellulose, which is especially weird because, like, that's plant stuff. Cellulose is cell walls, man. It's normally restricted to plants. A new discovery in sea squirts is that they appear to have primitive cousins to neural crest cells seen in more advanced chordates appearing later in time, in evolutionary time. So it would appear that these are potentially related to the precursors that produce the neural crest in more advanced chordates. So that's pretty cool. They just turn into pigment cells in the sea squirts, such that they have really pretty colors. So, I don't know. Neat. Okay, next we have Clade craniata. So, this is not a Linnaean classification. This is a clade. And craniata, craniates, are chordates that have a head with a brain. This is our big thing. Craniates have a head and a brain. During development, neural crest cells migrate from that neural crest throughout the head, and they can form other parts of the skull. For instance, they form uh, teeth. They form some of the bones of the skull, and they can form neurons and small sensory structures that are distributed throughout the body, which is kind of cool. So here we can see they have migrating neural crest cells in green, and then in this fetal human skull, you can see the portions of the skull that those migrated neural crest cells gave rise to. So that's pretty cool. Not only do we create the hollow nerve cord with the neural crests, but we get development of a lot of the features of the cranium. So neural crest cells are a huge feature of craniates. Craniates tend to have a much higher metabolic rate. So they have a higher metabolic rate. They're more active, higher metabolic metabolic rate, which allows them to be more active, more energetic. They tend to have a distinct heart, and that heart has 2 or more chambers in general. You're talking at least an atrium and a ventricle. Some of the simplest aquatic craniates just have a heart that's made up of a single atrium and a single ventricle. Right? Craniates have closed circulatory systems. Closed circulatory systems, which means you have the pumping heart and then blood vessels completely enclosing all of the blood. So there's no blood sinuses, there's no mixing of blood around the organs. We also have a complete digestive system with a mouth and an anus. Often it's a complicated digestive system with accessory organs, and the digestive system has muscle associated with it. So food is transported and metabolized through the digestive system via muscular action. And lastly, craniates have kidneys or nephrons that do the filtering. So their closed circulatory system connects up to the nephrons, the kidney organs that filter the blood. So that's how you get water concentration regulation and waste extraction in most craniates. Okay, so now that we've described the general features of craniates, let's get into some specific craniates. First off, we have superclass Agnatha. A is Latin for without. Nath is going to refer to jaws. Without jaws. These are what we call the jawless fish. Right? They do have tooth-like structures, but they are keratinized. So I'll put teeth in quotes because teeth are bony structures. So what appear to be teeth are keratinized structures. And these are generally for things like penetrating skin and attaching. A big thing about agnathans is that they do not have paired appendages. No paired appendages. There are two distinct groups of agnathans. Class Myxini, the hagfish. Hagfish have a notochord, right? So hagfish, they have a notochord, and they tend to be deep water scavengers. Deep water scavengers. Now, hagfish are pretty cool because they secrete a defensive mucus. And I got a nice picture of a hagfish here. There we go. They secrete a defensive mucus. If you have a bunch of hagfish in a bucket and you pull one out and mess with it, well, not pull it out, but poke it and mess with it, it will very quickly secrete a mucus that will turn all of the water in the bucket to gelatinous slime. So they can turn the water around them into this gelatinous slime as a defensive uh, strategy, I guess, to prevent something from actually being able to easily bite down on them. Next, we have the lampreys. Lampreys do not have a notochord. Instead, they show vertebra. So they have vertebra with the pads between, and their vertebra are made of cartilage. So our notochord has been reduced to the pads between vertebra, and we have cartilaginous vertebra. So lampreys belong to a clade that we refer to as vertebrata. And of course, this is a clade. Clade vertebrata. So, that vertebra has the spinal cord running through it. So we now have a protective structure for our dorsal hollow nerve cord. There are parasitic lampreys and non-parasitic lampreys. So if you've ever looked at a shark and seen those black eel-like fish swimming alongside them that don't appear to have any kind of paired appendages, so remoras or fish, sometimes you have a black eel-like fish, and it will attach to the skin and feed on blood. So let me erase some stuff to show my pretty scavenger lamprey here. So this is a lamprey that is scavenging, and there we go. So they have generally a mouth disc, and then on that disc they have these keratinized pointy structures. So in the parasitic lamprey, they use those keratinized pointy structures to attach and feed off of them. Marine lampreys actually have freshwater larvae, which is kind of cool. So there's our oral disc with our keratinized tooth-like projections. Okay, so that's superclass Agnatha, the jawless fish. Everything after that is what we call a gnathostome. Gnathostome. A gnathostome is characterized by having jaws. Okay, so gnathostomes are vertebrates with jaws. So if you have a jaw, you have a vertebral column. All gnathostomes are vertebrates. So, jaws are kind of fascinating. There was likely a duplication of a Hox gene that led to the evolution of the jaws. So you had a Hox gene. Remember, the Hox genes are these genes involved with directing what cells go where. The Hox genes direct what cells become a head, what cells become the mid-body, what cells become limbs. Right? So, uh, jaws are pretty cool. Um, the evolution of jaws is likely what allowed for enlarged forebrains and even lateral line systems. So that Hox duplication provided for a number of different features. Now, jaws are pretty cool. They are hinged structures. This is likely not amazingly new knowledge for you. And their primary purpose is, of course, feeding. So hinge structures for feeding. Before that Hox duplication, we likely had what would become the jaws simply being skeletal rods. So if you've ever eaten fish, you're pretty familiar with skeletal rods throughout the meat. So you had these skeletal rods for support of the gill arch. And there was a Hox duplication, and it caused some of those skeletal rods to uh, develop into something new. So, teeth are a product of those migrated neural crest cells, and teeth are likely something that are related to scales. So, for instance, shark teeth closely resemble shark scales. So much so that we refer to them as denticles. You might be aware that dent is generally a word related to teeth. These shark scales actually have the protein dentine on them, and they even have enamel covering them. So shark scales are extremely close to the structure of a tooth. It's really cool. So speaking of sharks, let's talk about sharks. Clade chondrichthyes. If you see ichthyes, you are talking about fish. Okay, so ichthyes. 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 Fish. Probably not a huge realization for you. Chondra. Chondra is the Latin prefix that refers to cartilage. So these are the cartilaginous fish. What does it mean to be a cartilaginous fish? That means that your skeleton is made of cartilage. Cartilaginous skeletons are pretty cool. They are extremely flexible. Extremely flexible. Allow a lot of these animals to be extremely agile. So, these cartilaginous fishes can be very agile due to their flexible cartilaginous skeleton. These were the dominant ocean-going predators between 360 and 280 million years ago. That means for 80 million years, cartilaginous fish were the dominant predators of the oceans. But they're actually severely threatened now. Hooray, humanity. So, uh, they have paired appendages, paired fins. So, in general, when you have paired fins, you have a pair of dorsal fins, right? The other ones that are easily recognizable are the paired pectoral fins. You also have paired pelvic fins. And so these are all for generally maneuverability. Pelvic fins are good for steering. Dorsal fins are good for maintaining position in water. They prevent rolling. Right? Buoyancy, the ability to, well, buoyancy is a measurement of how well something floats in water, right? So shark buoyancy, they do not reach what we call neutral buoyancy. Neutral buoyancy is where the animal's buoyancy equals the buoyancy of the water around them. And thus they can move freely in the water column very easily. Sharks and cartilaginous fish do not get to neutral buoyancy. They all have some degree of negative buoyancy compared to the water. In other words, they sink. This is because they do not have any specialized structures for buoyancy. Their buoyancy is provided by the liver oils. So livers in sharks or in cartilaginous fish are massive. They can take up to 2/3 of the internal body cavity. It can just be this massive oily liver. So one of the reasons sharks constantly have to move, why all chondrichthyans constantly have to move, would be that they will just sink, right? A lot of ocean sharks, like great whites and like I think black tip sharks, various ocean sharks, blue sharks, if you hold them still in the water, they will suffocate because they have to keep moving to keep water flowing in through the mouth and out across the gills. There are a number of different chondrichthyans that live on the sea floor or in shallow water. Shallow water or bottom dwelling chondrichthyans tend to have adaptations that allow the jaws to pump water. The jaws pump water across the gills. So, if you've ever seen a resting shark just opening and closing its mouth, it's actually pumping new water across its gills so it doesn't suffocate and die. So that's pretty cool. There's a large degree of variation in these animals, and especially when it comes to reproduction. I love chondrichthyan reproduction, shark reproduction. So let's talk about it. Okay, so one, sharks practice internal fertilization. So they have a cloaca, which is a common opening. A cloaca is a common opening. What does that mean? That it's an opening for both the digestive tract, the anus opens into the cloaca, and the reproductive tract. So, urinary, digestive, and reproductive tracts open into the cloaca, a common opening. So, male sharks have what we call claspers, which are specialized fins. So these are reproductive. And these claspers um, are basically sperm transfer fins. So you can see here, they will insert a clasper into the cloaca of a female. And then sperm will enter her reproductive tract, and then we will get fertilization of the eggs. And this is where things go crazy. So, right off the bat, if you've ever seen shark eggs, it's probably nurse shark eggs. Nurse shark eggs have a outer coating. Then you have a yolk and the baby shark developing within. This is called oviparous, egg laying. So it's actually very few species of sharks that lay eggs. You'll find these in shallow species, some reef species. But this is actually not the largest portion of shark reproduction. But you have egg-laying sharks, oviparous sharks like the nurse shark. Then you have ovoviviparous sharks, right? An ovoviviparous shark is right here. An ovoviviparous shark has an egg in the uterus. An egg develops in the uterus and then hatches. Then the egg and the yolk remain in the uterus. Well, not the egg, excuse me. The larva attached to the yolk, right? So the young hatch inside the uterus. I said larva. Man, I should just say young. There's no actual larva here. Oh man, come back here. Give me my pin back. So annoying. Okay, so ovoviviparous. They produce an egg. The egg hatches. And then the young and egg develop all within the uterus, all inside the uterus, such that the animal does what we call live birth. It gives birth to well-developed swimming young that come out of its birth canal and cloaca. So that's ovoviviparous. Then we have viviparous. Viviparous is pretty awesome. It's amazing that sharks are also viviparous because in viviparous sharks, right, the fetus develops with a placenta. So you have a placenta and there's blood exchange, or not blood exchange, but there's nutrient and gas exchange and waste exchange occurring between the mother's vessels in the placenta and the baby's vessels in the placenta. Just like humans, there are sharks that are placental sharks. So, chondrichthyes is just extremely cool. And we'll go ahead and end this lecture here and pick up with osteichthyes, the bony fish. So, there we go.